Good afternoon. My name is Bill, and I'm from the Graduate Institute of Biomedical Engineering at National Zhongxin University here in Taiwan. I would like to thank the organizers for giving me the opportunity to give this presentation, and I would like to also apologize for not able to give this presentation in person. However, if any of you have any concern about the content in this presentation, you're welcome to contact me through this email. The work I'm about to present are all based on this recent paper that we published in the Journal of Control Release, which is published this year in June. So basically, my group works on drug delivery, and for many of you probably aware that a lot of time the drugs that we develop or the nano detectors or fluorescent particles that we develop often don't end up at places that we wish them to. And that is because today's drug delivery still relies on what we call the EPR effect. Unfortunately, the EPR effect only able to enhance the delivery efficiency by less than twofold, as this paper pointed out. So in the context of tumor, you can imagine this is the reason why a lot of patients with cancer often suffer from severe side effects. That is because the drug that were administered into their body, hardly any of them reach to the tumor site. In fact, majority of the drug that were administered to them end up in organs like liver or kidney. Thus, these patients often suffer from severe side effects. So there is a landmark paper published in Nature Review Materials in 2016, which is still applicable today. And this group of authors did a systematic analysis on several clinical trials. You can see, regardless of which cancer types you're talking about, the drug delivery efficiency in those cancers are in fact less than 1%. What that means is that for every 100 drugs that you injected into those patients, only less than one of them actually reached to the target site. So it is pretty much, this figure had outlined that today's drug delivery is in fact not very efficient. So what my group had decided to work on is that we decided to see whether we can come up with a different delivery strategy, as in whether we can boost up drugs delivery efficiency to tumor microenvironment. So one thing we noticed, in fact, a lot of people have pointed out, is that when cancers start to develop, when patients start to develop tumor in their bodies, one thing that will happen is the recruitment of monocytes. What that means is that as the tumor starts to grow, the monocytes, which are one of the immune cells in your body, will come out from the spleen or the bone marrow. Now, once they enter into circulation, these monocytes have very strong homing capability for tumor microenvironments. In other words, they almost have their own GPS guidance system, if you like. So once they enter into the circulation, they immediately know where the tumor locates, and they will move towards the tumor. And the reason or the biology behind it is simply because as the tumor starts to grow, they will release a lot of chemoattractant into the circulation, and those chemoattractant were able to guide the monocyte migrate or moving from the circulation into the tumor microenvironment. So in other words, this homing capability or this recruitment of monocytes to the tumor microenvironment is very specific. Thus, so our strategy is very simple. Given that the monocyte will appear in the circulation as the patient starts to have tumor development in their bodies, we then will design nanoparticles or drug carrier with the anti-cancer drug inside, And the unique thing about our drug carriers is that they were able to hitchhike onto the circulating monocytes. So what I mean is that you don't have to draw the monocyte out from the circulation. Instead, you can just inject the nanoparticles with the drug inside into the circulation of the patients, and these drug carriers will immediately recognize the circulating monocyte and able to hitchhike on their surface. And they will remain on the surface of the monocyte, right, for at least 4 hours, which will show you the data. And this monocyte, given that they have very strong homing capability for tumor microenvironment, then will be able to carry the drugs, infiltrate the tumor microenvironments. And for those knows about monocyte biology, once monocytes enter the tumor microenvironment, they will become macrophages. Once they become macrophages, they will uptake the drug carriers that will anchor on their surface. And once the drug carriers enters the macrophages will break them down, and drug release will happen. And you will then get anti-cancer effect, which I will show you the data later. So that is the overall strategy of our drug delivery systems. So this is the design of our drug carriers. We call them monocyte-mediated drug carriers. The core is liposome, and you can have hydrophilic anti-cancer drugs or hydrophobic anti-cancer drug, depending on the chemical entity of the drug of interest. But the unique thing is that it has a PEG conjugated on the surface, as well a peptide that will specifically recognize a receptor on monocyte. So it's a receptor that's unique or found abundantly on the surface of circulating monocytes. And one thing I need to point out is that the ratio between the length or the size of the PEG and the peptide length is very important. So we did a very simple assay first to point out that our drug carriers can remain anchored on the surface of monocytes. Now this is very important because if the monocyte uptake the drug carriers upon contact, you will get immediate drug release in circulation, which become meaningless because that will be you'll get the same effect as free drugs. So it is important that our drug carriers remain on the surface of monocyte upon the contact with the cells. So the assay we did is very simple. So this is a static environment where we just expose the monocytes. So the monocytes are suspension cells. All right, so they're not adherent. They're in fact floating in the media, and we just add our drug carriers, which are fluorescent label. And after 2 to 4 hours, we centrifuge them to remove the excess drug carriers and have a look at them under confocal microscope. So if the drug carrier is able to anchor on the surface of the monocytes, as you start to scan the cells along the z-axis using the confocal microscope, what you will find is that the drug carriers are in fact encircling on the surface of the cell. So they're not inside the cell, but they're like forming like a circle. All right. So basically encircling on the surface of cells, so on the membrane, and not being uptake. And to demonstrate this interaction is specific, you can see that it only interact with the monocytes, which is THP1, and they do not interact with the endothelial cell. And this is very important because if our nanoparticles or drug carriers have strong interaction with the endothelial cells, that will lead to severe clotting effects. So this assay demonstrates that our drug carriers only recognize monocyte, but not the endothelial cell in the circulations. The next thing that we look at is whether the drug carriers that anchors on the surface of the monocyte could be internalized after cell differentiate into macrophages, just like they would in the tumor microenvironment. So as shown in the figure here, you can see while the cells are in the monocyte form, our drug carriers remain on the surface of the monocyte. But after we chemical treat them to then become macrophages, those drug carriers that were originally anchored on the surface of the cell will immediately internalize. So this shows that the interaction of our drug carriers, in fact, is a cell state dependent interaction. So well, I just went through all the static interaction. The next thing you probably think, all right, static sounds great, but in reality, the monocyte in our circulation is a dynamic um, response or dynamic phenomenon. So what we did was, the next thing we did was in fact set up this microfluidic device to mimic blood circulation. So we first inject the monocyte, let them circulate for a while, 37 degrees. Then we inject our drug carriers into the microfluidic device. After we let them cold circulate for about 2 to 4 hours, we start to take time-lapse images. So if you look at panel B here, you can see that our drug carrier showing red was able to hitchhike onto the circulating monocyte, just like it would in static. The control group are the drug carrier formulation that we now use in clinics. So this is the PEGylated liposomal formulation that we use in clinics. And those clearly have no interaction with the circulating monocyte. And if you extract the cells out and do the flow cytometry analysis, you can see that our drug carriers can in fact successfully able to hitchhike onto circulating monocyte even under very high shear stress. So the interaction is in fact very specific and has high affinity. Okay, so after we showed that our drug carriers can able to hitchhike onto circulating monocyte, the next thing was we use the microfluidic devices to establish a 3D culture model, try to recapitulate some of the key characteristics we see in tumor microenvironments. So we first established an assay where we have the collagen gel to mimic the ECM or the tumor microenvironment, and on top of that we have a layer of endothelial cell. Then we inject the monocytes and with the drug carriers. And after it circulate for a while, and since because the endothelials are not inflamed, you can see not many monocytes attached to the surface of the endothelial barrier. But yet those attached still have the drug layer on top. However, if you induce the endothelial barrier to become inflamed, just like they would in a tumor microenvironment, the monocyte will in fact adhere to the inflamed endothelial cell very quickly, and it's showing here in the figure down here. And those monocytes will also carry the drug carriers at their surface, but most importantly, as shown in figure E here, those monocytes will undergo in extravasation or transendothelial migration into the gel underneath the endothelial barrier. Okay, just like they would in a tumor tumor tissue. And if you do some acid analysis, you can see that if you stimulate the endothelial barrier with TNF alpha, it will excrete more NCP1, which are the key chemokine that attracts or recruit monocyte to the inflamed tissue, and that the amount of the adhered THP1 is in fact quite high when the endothelial barriers are in fact inflamed. So this shows that our model in fact was able to capture several key physiological conditions that you see in patients with cancer. All right, so you probably now thinking, all right, that's the studies or the results show is in fact all chemical treated. Is there more? Is there more like more physiological relevant? So what we did next is in fact instead of chemical stimulate the endothelial barrier to become inflamed, we decided to let the cancer cells do the job, just like you would expect in a patient with cancer. So in the collagen matrix, we in fact have the MDA MB231, which is a human breast cancer cells. Then form several tiny spheroid. Then we have the endothelial barrier established on top. Then we inject the monocytes, followed by the drug carriers. And these are the results. So you can see that once the cancer fibroid starts to grow, then we induce the endothelial barrier to become inflamed, and you got all these monocytes will try to attach to the surface. And so again, if you figure A, just a screenshot of the video we just saw, And if you look at the cancer cell in the gel, or the spheroid, which is showing panel B representing blue, you can see the several spheroid established. Most importantly, those monocyte was showing green. After they are here, they were able to transmigrate from the barrier into the gel. And if you took a enlarged screenshot of any of these infiltrated monocyte or monocyte-derived macrophages, you can see our drug carrier, which showing red, was able either remain on the surface of the cell or being internalized as the cell has start to differentiate into macrophages. So this shows that our model, in fact, was able to recapture some of the, or basically this model is more clinical relevant to the tumor microenvironment that we're seeing in cancer patients. Now, to demonstrate our model is in fact tumor specific, we established another microfluidic in vitro model where we replace the cancer cell with a non-cancer cell line. In this case, HIG293T. Again, we have the endothelial cell, endothelial barrier established on top, and with the monocyte inject the monocyte followed by the drug carriers. I can show it in the video here. Showing the video here, you can clearly see that since the HIG293 T cell are not cancer cells, therefore they cannot secrete any cytokine that could induce the endothelial barrier to become inflamed. So you can see even if you inject a lot of monocyte across those endothelial barrier, none of the monocyte in fact adhere to the endothelial barrier, since the endothelial barrier were not inflamed. So that's why you have no or very little monocyte adhesion happening. So again, this is a screenshot of the video we just took earlier. You can see that the green is the monocytes. You can see not many of them have attached to the endothelial barrier since the endothelial barrier are not inflamed. The blue are the HIG293 T cell. Again, it can just show that the cell survive very well in the matrix from this 3D culture. And panel C meant to be the signal of the transmigrate monocyte, which no longer occurs since very little adhere to the endothelial barrier, so there was not enough signal to trigger them to undergo transendothelial migration. So I just show you that the in vitro model demonstrating our drug carrier is very specific. It's only specific to tumors microenvironment, and we are able to use the 3D microfluidic in vitro model to demonstrate that. So next thing is this is the final slide for the data, which is the in vivo data. So this is some of the key data, so I'll show you quickly. So again, we established a mouse model with a human breast cancer, and we just inject the human monocyte and then circulate for a while in the mouse. Then we inject the drug carriers, and the control is the PEGylated liposome. And after that, we extract the monocyte out from the circulation and did a flow cytometric analysis. You can see that the hitchhiking efficiency is about close to 50%, whereas the control group is only about 2%. In terms of tumor targeting efficiency, with the above distribution assay on different tissues, you can see that the blue colored bar representing our drug carriers is in fact 10 has tumor targeting efficiency that is tenfold higher, so it's tenfold higher than the control group. And with the simple therapeutic efficacy analysis, where we capture doxorubicin, which is an anti-cancer drug that's right now commonly used in clinically treating breast cancer, and capturing our drug carriers. And if you give a single shot, you can see that it's able to shrink the tumor much more better compared to other control group. And this histology staining just show that our drug carriers were able to boost up doxorubicin activity to induce more activated caspase 3, which lead to apoptotic effect of the tumors, and able to downregulate the proliferation as well as their chance of undergoing metastasis.
So this is my final slide. So this is a summary to show that we established drug carriers and able to hitchhike onto circulating monocyte. And the reason for that is because the monocyte have very strong homing capability for tumor tissue. In other words, once the patient starts to develop cancer, the monocyte will only go to tumor tissue and not the healthy tissue. Thus, we harness these physiological effects and allowing our drug carriers able to hitchhike onto the monocyte, so you don't have to extract the monocyte out. Now drug carriers are able to hitchhike onto the circulating monocyte, and the monocyte will carry the drug carriers with the drug to the tumor tissue. Once they become macrophages, will uptake the drug carriers, break them down, and drug release, and the drugs will then kill the tumor tissue. That's it from me, and I thank everyone for listening. Thank you.